Hello, everyone, and welcome. My name is Eleonora Chegaray. I am ICTA's Marketing and Communications Director, and on behalf of the team, I would like to thank you for joining us today for the webinar, AI Orchestration in Drug Discovery, from de novo design to automated synthesis. Before we start, let's do the usual housekeeping. Throughout the presentation, we will be sharing relevant links and content in the chat, so please make sure to review the chat function at the bottom of your screen. To ask questions, please use the Q&A feature, and you can do so anonymously if desired. Questions can be typed in Japanese, as we have many attendees from Japan, and they will be translated for the speakers to answer at the end of the session. The webinar is recorded, and you will receive the link via email within 24 hours. Please feel free to share the link with colleagues or relevant contacts. So now let's introduce our speakers for the day. We are joined by Frank Levaillant, Senior Scientist and Deputy Head Lab at ICTOS. Frank has a master's degree from the École Nationale Supérieure de Chimie de Montpellier. He carried out his studies at EPFL, his PhD studies, in Switzerland, under the supervision of Professor Wasser, during which he explored photoredox catalyzed functionalizations of radicals using hypervalent iodine reagents. Then he moved as SNF postdoctoral fellowship in the group of Dr. Cornella at the Max Planck Institute in Mülheim an der Ruhr, where he developed nickel-catalyzed CO bond formations using nitrous oxide. He returned to France to start the LabCom HitCat, a joint venture between the company Sequence and the group of Professor Masson at CNRS. Since July 22, he has joined ICTOS as Senior Scientist, Deputy Head Lab. He works on the installation, development, and utilization of the ICTOS robotics platform for AI-driven drug discovery. Thank you, Frank, for joining us today. Now, the webinar will be opened by Quentin Perron, ICTOS Chief Scientific Officer and co-founder. Quentin is a medicinal chemist by training, and he holds a PhD in organometallic chemistry from the University of Geneva. During his postdoctoral fellowship at UCLA, he worked in the total synthesis of brasilicardin A, a complex natural molecule known for having potent immunosuppressive activity. After working as a medicinal chemist in CNS indications at Laboratoire Servier, he switched to data science and chemoinformatics at Quinten, a company specialized in data science services. In 2016, together with his business partners, Jean-Gaston Maté and Nicolas Dohu, he co-founded ICTOS. As CSO, Quentin brings his vision for innovative AI technologies to accelerate drug discovery, with solutions ranging from the novo drug design to AI-orchestrated robotic synthesis, which we will be discussing today. Thank you, Quentin, for joining us, and the floor is yours.
Thank you, Anna. Good morning, everyone. Good afternoon. We are very pleased, Frank and I, to present you what we are doing at ICTOS, how to go from a listing of molecules designed by AI to the synthesis of those compounds onto an automated platform. So today, the agenda is quite packed. So fasten your seatbelt and let's go directly to the presentation. So you know that going from a target to a drug candidate, it's something which takes a lot of time, which costs a lot of money, and the figures are not very good. 50 to 100 million euros, 3 to 5 years, thousands of molecules to make, and a very low success rate. There are two main questions you need to ask yourself in this endeavor is, what molecule should I make and how to make it as fast as possible? So right now, those molecules are designed and made by chemists. This is a traditional way of imaging molecules and making molecules. And we asked ourselves 2 years ago that, is it possible somehow to incorporate somewhere in that process some robotic solution? The goal was not and is not to replace the chemist, but it's really to try to accelerate that process, to find some bottleneck where we could put a robot to accelerate that. And here you are the challenges. One of the main challenges you need to ask yourself if you want to build such a platform is, first, what type of molecule are we talking about? We're talking about of very simple structures with very simple chemistry, Are we talking about complex singleton or complex library in a few steps, starting from advanced intermediate or somewhere in the middle? The second main challenge you need to ask yourself is what type of chemistry on your platform? What do you want to run? The more the chemistry, you will have freedom on your chemistry, the more you will be able to go from a library design, very static things, where you have simple starting material with very few positions you can modify, to drug design, where and rational drug design, where you can modify the right hand side, the left hand side, and you have a lot of freedom in terms of chemistry. The third main challenge you need to overcome is organization, how to make decisions. Let's say that you have 300 molecules you want to make, all different, from simple to complex structures, from simple to complex chemistry. If you want to do that, you need to ask yourself for each of those molecules, what synthetic routes, what starting material, how to communicate easily with my providers to place orders, how to fill my ELN. I didn't receive some building blocks. What should I do? Should I wait? Should I discard these molecules? Which reaction can I launch in parallel? So you have so many questions you need to figure out in order to be able to treat a huge listing of molecules. Finally, the fourth main challenge you need to overcome is what type of robots should I consider? Should I consider flow chemistry, batch mode, how to transform a protocol that I can find on Reaxys or on US patent database into a robot instruction? And who is doing that? Is it AI? Is it scientist? Is it the chemist? Who's going to launch the robot? How can I track my viral position to avoid mistakes, to ensure that the robot is doing what I'm asking, what I want it to do? How to handle different chemistry, solvent, reactant, reagent, etc., etc. So also, being able to put chemistry on the robot is something difficult. So what we have been able to observe is that overwhelmed by all those challenges, the chemist made it simpler, basically. So usually when you see robotic platform, people show that you can do only simple molecules. The chemistry is very limited. You do one reaction at a time, and very often the workflow is quite encoded and not very versatile and dynamic. So what we can observe is usually you have a core scaffold, you have an exit vector like for here, aniline, and then you have a list of, in this example of amide coupling, you have a list of acid moiety you want to branch on that nitrogen. This type of workflow and setup works pretty well on a robotic platform. It's very simple, the same reaction in all the wells on the robot. The problem of that type of approach is that we call that library design. The impact is very limited because in real life, it doesn't work like that. You don't want to do a library design in a given position, maybe at some point in your project, but just for 10 or 20 compounds, not for 100 or 1,000 compounds. So this type of approach, there is no adoption. And we have this dramatic example where the Lilly platform, they have been able to build an amazing platform, $90 million spent to build this fantastic platform, but now it's dismantled because those type of platform are too I would say static and allow they are not very versatile. So what we want to do at Ictos, and we ask ourselves, are there new ways we can use robot today? Because we don't want to do library design. We want to start from a very different molecules, different type of chemistry. I don't want to think about my chemistry. I just want to tell to my system, I want to make those 200 molecules with a different chemistry, different exit vectors. I want simple, complex structures. So I really want to go from library synthesis to rational drug design in a totally automated workflow with a nice and user-friendly interface tailored by chemists for chemists. So if you want to be able to do that, and if you want to be able to overcome those challenges, for the first challenge, you need to be able to produce a list of molecules which are feasible in the robot. Second, you need to make sure that for all those feasible molecules, you find routes which are compatible with your robots. Third, you need to select the best route if you have many routes for each molecules. And finally, you need to be able to execute that process on the robot. And to be able to do that, you need to break the silo. You need to make sure that all those technology are communicating together, because if you imagine a fantastic molecules, which cannot be produced by your robots, forget about this platform. If you produce a molecule which is feasible theoretically on the benchtop, but not on the robot, it's the same. You can forget about your platform. So you need to make sure that everything is connected together. And for that, it's difficult for a chemist to think about at the same time the molecules you want to make, but also to take into account all the constraints of the system. This is why AI can help. And we have developed since 2016 at Ictos many different types of technology: Maca to generate molecules, Spire for the retrosynthesis, Ilaka for the workload organization, and Ictos robotic to execute that chemistry. So if we go to Maca very rapidly, Maca is not a virtual screening approach. It's a generative AI approach, which has not been trained on smiles representation like everyone is doing, but it has been trained on organic reaction. So by design, Maca generates feasible and novel molecules. We can explore an enormous chemical space estimated to 10 to the power of 27. And of course, it's not a random exploration. It can be guided by 2D QSAR models, presence, absence of substructures, 3D structure-based core, bolts, docking, whatever you think of. Of course, it's very easy to use. And also, the molecules we are generating are compatible with the robotic constraints. So we already presented that recently in our previous webinar. So you can just scan the barcode here, scan the QR code here, and you will be redirected to the video, the content, the benchmark, the papers, and everything you need to know about Maca. The second technology we have developed is called Spire. It's for retrosynthesis. Like Maca, Spire has been trained on organic reaction, but contrary to Maca, which starts from building blocks and reaction to imaging new molecules, Spire is doing the reverse. You put the molecules and try to find reaction and building blocks to make these molecules. Spire is data-driven. On the next slide, please. Spire is data-driven. So you give a database of reaction, you give a database of starting material. In less than a week, we can retrain Spire. Right now, Spire is publicly available on spire.ai. It has been trained on US patent database. We have also trained Spire on Reaxys data, and you can have access to this technology through the Reaxys platform as well. Spire is quite unique because it has plenty of option where you can play on the price of the starting materials, the delivery time, the providers, but also the name reaction. You can force Spire to pass by a certain type of name reaction, avoiding a different type of chemistry. And this is the cornerstone of the robotic approach, because thanks to Spire, you can triage molecules which can go to the platform, the robotic platform, because those molecules are passing by the chemistry you master on your robot, but also filter molecules which are going to go to external CROs. You can also define building blocks, key intermediates, presence, absence of substructures, or even some bonds you want to protect or you want to cut during the retrosynthesis. Spire is available through Spire GUI if you are a chemist, you want to explore the roots, or through Spire API if you want to plug this technology to an external system. And this is what Frank will be able to show you, how Ilaka is organizing the workload, transforming a list of molecules into a list of instructions for the robot, calling Spire API at the, in backend.
Okay, so yeah, I will show you how we developed a new technology that is called Ilaka, which is based on a new software technology called scheduling AI. The idea behind Spire, behind Ilaka, is actually to make a rule selection and compound ordering and to try to make it easy. So in Ilaka, we are able to develop a new reaction type and also to set up campaign of reaction for hundreds of reactions. ILACA integrates the proprietary scheduling AI, and we try to organize the workload that the chemist will do in the lab. And we also try to facilitate, especially for small company, a way of actually placing order and buying building blocks. So we can directly use ILACA to directly drive the robot and try to limit the human intervention in the lab. And very importantly, we try to deliver it through a user-friendly interface, because otherwise the adoption in this kind of software will be very limited. So after getting the roots from Spire, very importantly, it's how we can translate this information into something that is readable by the robot. So at Ictos, we try to make some AI-driven extraction from the patent procedures. And it was possible through natural language processing to actually get all the information required to be run on a robot. But however, we were wondering a few questions that were still remaining. And the first one is actually, is it enough? Do we trust the patent protocol or the data that is inside of this patent? Also, are we sure that the AI extraction will be 100% reproducible? And maybe sometimes the lack of generalized way of writing a palladium catalyst may hamper these extractions. And also, is this extracted procedure is compatible with the robot? And this is a very important point, because sometimes, and usually all your robots have constraints and you need to adapt it. And translating something that has been done on a patent procedure in the lab in a manual way, it's difficult to directly transfer it in the robot. So we were wondering how we can try to enhance this. And actually, this is a very high topic so of how we can get a reaction condition. And this has been even discussed by Korean coworkers later this month in Chem Science. So in our case, we actually decided to use expert-based templates. So to develop our own chemistry that is compatible with the robot. So in this case, we know the constraint of our robot, and we know how to apply chemistry that is actually compatible with this. So we avoid the low temperature, so minus 78 reactions are absolutely prohibited. We try to avoid using very reactive starting material or hygroscopic starting materials that will hamper the delivery of the chemicals. And thanks to Spire, we are able actually to use only reactions that will give a single radioisomer. So we did this very important work beforehand, before setting the platform. And actually, it's something that we're still continuously developing. It's an expert-based approach, and we use literature data. And actually, we're also introducing new way of doing chemistry. So we're introducing batching and clustering parameters. So as for example, here on the Suzuki, we will set a time and a temperature range where we think this condition will be applicable. So for example, the condition for Suzuki version 1 will be 60 to 80 degrees with a special palladium catalyst and a solvent, and we can apply it during 4 to 8 hours. We will also embed inside actions, so we can say that there will be no quenching, there will be filtration and a final dilution. And we will do the same for any name reaction we are applying on the robot. So far, we have developed more than 65 name reactions, and we are always continuously working on that. So in ILACA, the way we can work is actually to develop new name reaction, and we can register all this information in the template. Here you have the example of how we define it. So basically, you have the name reaction, you will have the name of the template. You can define the reactant and reagent you want to use. You can define the solvent and the concentration of your reactions and the key robot actions. So everything will be already embedded and pre-filled later in ILACA. All this information is so far saved as a new reaction template. Currently, we have 65 name reaction in the lab, and this corresponds actually to 130 templates. So we try to make several versions, like a Suzuki 1, Suzuki 2, to make it more flexible in the robot. This will also help us to make a better batching and clustering approach. Among these 65 name reactions, we are already mastering most of the palladium catalysis, so Suzuki, Negishi, Hex, Nagashira coupling, but also very importantly, the Buchwald big amination. We also perform copper catalysis, ester and sulfonamide bond formation. Obviously, SN2 and SNAR are common in our working way. And we recently developed some oxidation and reduction reactivity along with protection and deprotection. Sometimes in our project that we're working on the pipeline for medicinal chemistry, we also need to make actually heterocyclic chemistry, and we already developed few new name reaction around this. So I will show you how we actually work at ILACA. The very important aspect is how to transform this list of molecules that you may have received from your drug design, and then how to transform it in a procedure that will be readable by the robot. So basically, we upload the list of molecules that has been designed either using Mica, that is obviously better because you take into account the synthesis constraint, or you can directly have it from other type of design. We'll call SPI API in the backend, and actually we will get a lot of synthetic routes that are robotic compatible, thanks to our filter that we are applying there. And the difficult part is actually how to make this selection to get only one route per molecule at the end. So we are actually applying some filter and some comparison where we can say that this exact route for the molecule 1 will be more compatible with the second one because they will share at some point a common intermediate, or they will share a common temperature and time, and they will be able to be batched together in the same reactor. So at the end of this process, we transform a list of molecules in a list of reaction, and we will have one route per molecule. And we will submit this to the chemist for review and validation. And if the chemist is not happy with what has been proposed by the AI, we can directly restart the loop. So a very important step is actually the validation by the chemist. It doesn't take a lot of time. It's usually 1 minute per reaction. So the process is submitting the list of molecules in the strategic request. We will define the retrosynthesis constraint. If we want to have 2, 3, or more steps, it's possible. We can also play with the building block price and a few other parameters. Then during the reaction review, it's possible to check that the name reaction has been retrieved correctly, and we can assign a template. As soon as we assign the template, we actually load all the information that has been embedded inside, and this will provide you with reagent, solvent, and other conditions. With this automatic filling, this is actually very fast, and the final step is actually to adjust the stoichiometry and the scale of the reactions. If the chemist is happy with his setup, he can just approve it, or otherwise he can reject and restart the route. Once the route and reaction has been validated, the chemist can go to the procurement system that we have building inside of ILACA, and it's possible to send automatically email for quotation and purchase order. So we are directly communicating with a provider, and the provider will actually fill the information, and the information is gathered back directly in ILACA. So everything will be embedded in ILACA and this interface. When we are receiving the pre-weighted compound in building block barcoded vials, we can directly load them in this smart fridge. So the Hamilton VersoQ Series 20. It's a smart fridge that actually makes a very fast reception. So we can scan all the barcode at once, and we can directly update ILACA. So as soon as we have building block reception in the lab, we can have the stock and the inventory up to date. And then we also develop Ictos Robotics. So it's based on the ChemSpeed platform. We have ordered and put in place a robot that is capable of running 96 reactions in parallel. And so far, we are using 65 name reactions on this platform. In less than a year, we are able to go through more than 3,000 reactions, and this is not our average speed. We can go for sure much better. We deliver more than 300 final molecules for the pipeline in drug discovery project, and this has been synthesized over an average of 2 steps. Currently, the success rate is around 60 to 65%, and we are always working on improving the templates to actually enhance the success rate. And it's possible that one chemist can operate one robot, and we are trying to work on the user friendliness on this approach. Here is our ChemSpeed layout. It's actually designed to translate our vision of organic chemistry in an automated synthesis way. So our robot can make 2 main steps, which are the inventorization and performing chemistry. So obviously, it's possible to hold and move vials, read all the barcodes with the barcode scanner that is here, We can make also a database operation that is indispensable for making this approach of clustering synthesis. We can obviously perform all the minimal step of organic chemistry, which is dispensing solid and liquids, heating up and cooling down, stirring, shaking, filtration, or evaporation. The ChemSpeed platform comes along with a software that is called AutoSuite. It's a very nice software where you can actually design a lot of parameters and you can execute exactly what you would like to do. However, on a daily basis, this is quite challenging. So it's really an expert-based software. And for example, moving a powder from the rack of powder to the solution or to the reactor, this is 16 parameters. And moving a substrate from the substrate rack to the reactor, this is more than 40 parameters. So when we are running it on a daily basis, this is quite challenging to hand code every time this program. So we wanted to simplify it. For us, the way to do it, actually, it was to try to design one program only. So a program that will actually be able to encompass all different type of campaigns we want to use on the robots. So we took all the constraints of our robot inside, and we parameterize all the different functions. We put this in what we call macro, which is actually a program, a functional program. And we can make database reading. We can open different parts in the robot. We can create zone and we can transfer materials. And when we do this, actually, it's possible to iterate or go to the next macro based on the data that you will input in your program. So we have this program plus a series of CSV files or a database that actually will be compatible together. All this data that is readable by the robot then allow you to have only one program going from adding powders to heat and stir, and at the end going back to the initial state. So we succeeded in making only this one program, and now the chemist doesn't need to program AutoSuite anymore. The way we run ILACA is actually to try to be able to make multiple projects on multiple exit vector in the same reactors. So this is an example where, for example, you could have 3 drug discovery projects running simultaneously, 96 molecules that are ready to make, and you have 3 projects with 6 different name reactions, different exit vectors also. And the way we will push it in ILACA actually is as it is here. We have the 10 Bromo Suzuki, the 28 Bromo Buchwald, and the 10 SN12 chelations that can be combined together in this first reactor of 48 reactions at 80 degrees for 10 hours. And we believe that actually the reactor 2 could be involving the 20 amide bond formation, sulfonylamine bond formation, and Mitsunobu reaction at room temperature overnight. In this ideal situation, this is how we will execute the campaign, and we will show you later some results like this. When we are ready for launching this daily campaign of reaction, it's very important that all the information is shown to the chemist. So we actually deliver an action planning in ILACA, where the chemist can have all the information on the structures, the barcodes, the quantity, and where they will be located in the robot. And then we can push this information and load the campaign through ILACA. So overall, when we are operating the ChemSpeed apparatus, the chemist will load the robot. Once the platform is ready, he can push in ILACA the campaign. All the information will be sent through our database, that is a cloud database, connected to the computer of the robot. This will send execution step, and we also can get back information from the robot live. Everything will be sent back to ILACA at the end of the campaign and will be saved actually in our ELN. So what we have shown you today is actually that from a list of molecules, a list of smiles, it's possible to input this in ILACA. We will call SPI API in the backend, and this will provide you a list of reaction. From this list of reaction, it's possible to divide it in several ways. Either you send it to the CRO if you think that it's not possible to make it on the robot, so you have a possibility of fast rejection, or you can combine it directly to the hardware agnostic AI and automation that we're delivering now. So we are going through ILACA, and this will tell you how to get the procurement, how to organize your workload, and start the reaction in the ChemSpeed platform. We're also now connecting actually analysis and purification later on. So this is coming soon. And we also implement the connections through our ELN. Importantly, what we have found while running this workflow is actually during the heat discovery phase, 80% of the molecule can go through the robot and about 20% can go to the CRO. And this number is decreasing slightly after when we are reaching heat to lead and finally at lead optimization, 10% we are working on the robot and 90% on the CRO. So we believe that actually we are not replacing manual chemistry, but we try to make it faster, especially during the discovery phase. And I will hand it over to Quentin for the POC presentation.
Thank you, Frank. So yeah, we wanted to perform some sort of a stress test of our robot. So the idea was to perform a parallel synthesis of 135 completely different molecules. As you can see on the left, it's just an extract. And we have used our generative AI to do so using our in-house inventory plus our in-house reaction. And as you can see, those 135 diverse molecules can be made using 27 name reaction and using 231 commercially available building blocks. As you can see on the left, you have plenty of different types of chemistry, which can be run the same day on the same platform. And so we gave those 135 molecules to ILACA. ILACA, as Frank explained, automatically transform those list of molecules into 3 campaigns in ILACA, defined by 11 molecules for the first campaign, 29 for the second one, and 95 for the third one. Those campaigns has been split over the 2 reactors with different cluster of temperature from 10 degrees to 105 degrees. Overall, we obtained a 70% success rate, which is quite good in 1 reaction steps, with a purity going from 90% to 100%. And we are preparing a manuscript, and we hope it's going to be published before the end of September. We also tried this platform not only to test how different the chemistry can be run on the same platform the same day, but also to push a program where we decided to tackle the target PKMYT1 and using Maca and generative AI, Spire, and the robots to streamline this design, make, test, analysis cycle. In less than 3 months, we have been able to identify 4 novel and potent hit series going from 30 nanomolar activity to 800 nanomolar activity with a low molecular weight, very good LIPI, and a totally novel chemotype compared to other competitors in the field, which exhibit a special scaffold which make those compounds active. We found a totally different scaffold with a very nice activity as well. We are using this platform not only for our POC we just showed previously, not only for PKMYT1, but also for flagship program on the target called MTHFD2. And we hope that before the end of this year, we're going to have a preclinical candidate. Frank, the floor is yours to show us how the platform is running through ILACA. So it's going to be a demo of the technology.
Okay, so this is our ILACA. It's a software where you can directly make a single sign-on authentication. So you have your personal account. It's a collaborative environment. You have all these name reactions that are already embedded. We make the connection between lab name reaction and analogs. So analogs are actually the Pistachio name reaction from the Pistachio datasets that has been curated. We can link the name reaction to templates, as mentioned earlier. And we have all the detail in this kind of templates. So it's possible to get the information on the stoichiometry, on the different reagents you want to use. You may have all the various information that could be a link to equivalent of a base, concentration of your solvent. It's possible to make also actions directly inside, so you can select and unselect via some toggles the different actions that you will run on the robots. What is important is actually we can modify and implement more of this action, and this will depend on the hardware you may have. At the end, what is very important here in the template is to have the information on the time and temperature. So this is the batching parameters, and this is what will allow us to make the clustering of all the different reactions together in the same reactor. Okay, so we'll show you now how we make actually a retrosynthesis. So we go in the strategic schedule. You can get your list of smiles of various compounds. It's also possible, obviously, to make library design if you have a common intermediate, but we were able to actually work on different workflows. You select the project you want to assign all these molecules, and you submit by validating these smiles. It's possible after that to actually select the different name reactions. So you can either select all or select only a few of them. Now we actually put a setup features in place where you can make this preset like Inspire. So we can have already embedded a list of name reactions that we want to use. For example, the robotic constraint, the robotics reaction that we know obliging the robot constraint. If you have an intermediate, you can submit it. You can play with the maximum depth of your retrosynthesis and the price of your building blocks. It's possible to deal with radio issue and remove chirality directly if you think that this could be an issue later on in the retrosynthesis. And then you submit it. And as soon as it's submitted, it will call SPI API in the back end and make the high throughput retrosynthesis of all these 9, in this case, targets. We already apply it to 150 or 200 molecules in the same time. It may take a small moment, like 1 or 2 minutes for these 9 reactions. And usually it's relatively fast. I think for 200 reactions, it takes 1 hour or something. In the result, we can download actually a report. We have all the information that are the pistachio name reactions and the reaction smiles. We have a scheme. The report will tell you also the information on the success rate of your retrosynthesis. And it's possible to directly navigate quickly to the reaction that didn't pass this retrosynthesis step. You can find all the details in the reaction review panel. You have some filtering possibility where you can go directly to the ones that are pending. You can make this fast rejection directly with the red button there if you don't like this deconnection. But if you like it, you can process to the review. In this reaction review, you get back the information of the name reaction. It's possible then to change it if you think that actually it was not correct. And it's also possible to assign the templates. So here you will see that you have the list of different templates that will be shown up, and you can make the selection automatic filling of the reaction details. You can choose your limiting reagent directly here, and it could be possible also to adjust actually the scale. We are usually running at 0.1 millimole, but it's possible to go a little bit higher, but you need to stay within the maximum volume of your vials in the reactors. So currently in ChemSpeed, with our 8 meters vial, we cannot go higher than 4 milliliters in the vial. So the chemist is noticed if it's passing higher than this limit. You can adjust, obviously, the equivalent of your different substrates, and everything is automatically calculated. So it's very similar to an ELN. After that, we can approve the review, and we will need to make the ordering of these compounds. So either you have it possibly internally, so you can click in internal and you have a check of your inventory, or you can go through our new procurement system interface. So this is what we will show you today. Everything is saved in ILACA, so it's event-based software. So all the information will be saved in a database, and we have all the information on the quotation. So here we just show in this tab that we collect back this information. So if we go to quote, you will have some quote here with the compound that we wanted to have. And it's possible to just select one, or you can actually make a final selection of all the compounds and submit this quote to ChemSpace. As soon as this is submitted, an email will be automatically sent to the provider. And now we will be placed in the role of the provider, and this is what he will receive in his mailbox. It's a mail from Ictos procurement system with this link. And in this link, you have all the information necessary to make a good quotation. So they will get back the structure, the smiles. If they don't have this compound, but they have a solve, they can mention it, and it will be automatically calculating the new mass that is required. And what we are asking for the provider is to get the price and the delivery time of this compound. When everything is ready, they can submit the quote, and Ilaka will retrieve this information. So this is usually very, very fast also. And so it's allow us to actually be fully independent in this way. When you have the information that has been collected here and it's obeying the rules of retrosynthesis, everything is in green. So you can just approve your quote, but you can also reject your offer here if you are not happy with the price or with something like the compound they have. It could be like a different radioisomer or stereoisomer. Then you can progress with the purchase order. So again, here it's possible to make a maximum delivery time that you want. So if you want your compound in minimum, a maximum of 8 weeks, you can mention it. And everything again will be sent to the provider. So here again, the provider will receive this new purchase order by email. And in this case, you have the information and the provider will need to put the barcode and place the amount, pre-weighted amount directly inside of these vials. So what we will receive in the end is actually barcoded vials with 1D on the side and 2D on the bottom. And we will have this list of barcodes and delivery time submitted to Ilaka. Okay. So this is the end of the procurement system. And after that, we will make actually a reception in the lab. So you can see here in the inventory now these 2 building blocks that were following since the beginning are actually incoming. So they have been shipped by the provider, and we are recovering already the information, the barcodes, and the quantities that will be inside of these barcoded vials. When we receive it in the lab, we can directly take a Hamilton rack and submit it to the Hamilton, but otherwise we can write it manually inside and scan this container. So it's quite efficient even manually, and this is not taking a lot of time for us. Everything is updated instantaneously in Ilaka, and the inventory then is up to date. Okay. So once we are ready with all the compounds, what is possible will be to make a schedule of the campaign you can make today. So you can see that actually the compounds has been now passed in stock, and the reaction will be going to a new status. So they have been moved from schedulable now. So once you have many reactions that are schedulable, it's possible to create the schedule. We have 3 ways, either a manual way where I can just see what is possible to do, and you click on the molecule you want to see. And then you can see on the right that you can fill automatically your robot with the data for the campaign. But this is a really manual way. And then all the information is collected here. You have the powder, you have the solution, you have the solvent. And obviously, if you are going higher than one of these parameters, you will have a red flag. If you're happy with this, you can continue. Otherwise, you can also have another way of doing it. It's possible to organize the workloads through a different lab, or even if you want, you can choose which robots are available at your platform. So this is really a scalable approach. It's possible to make filtration through projects, and it's also possible to actually make some filtration through name reaction. And also, you can play with the temperature range by making a selection and removing the number of molecules you want to make today. So this is what we call the supervised reaction schedule, and this actually helps us to make very quickly a campaign. So if you think that today you don't have a lot of time remaining, but you think you can make quickly these 10 compounds, you make a batching like this. And finally, we have the master of all daily schedule. It's actually the automatic one, where Ilaka will just gather all the compounds that are remaining inside and the schedulable reactions that can be processed today. So for example, here, we had 6 reactions that were schedulable, and Ilaka proposes to put them together in the same 2 clusters. So we have first cluster number 1 with 5 reactions and cluster number 2 with 1. You can see the different name reaction that has been set up inside here. The temperature range were actually similar, but in the time range, you can see that there was 2 different types of time range, and Ilaka is making the intersection. It's possible to approve it, to decline or cancel all these schedules. And we apply this to the 135 reactions that Quentin mentioned it earlier in the POC. And this gave us actually these 3 clusters here with 2 batches for each. And we got the main reaction was a 95 reaction at the same time, with the cluster having more than 10 different name reactions inside of each reactor. When you're ready for making a campaign, you can download and push the instruction to the database. This is action planning, where you have all the information shown to the chemist with the barcodes and how it will be looking like inside of the robot. So this is really helping to go from virtual world to reality when you are in the lab. We also provide a tool that is actually helping us to make very quickly the stock solutions that are required to run the robot. So this is a very classical tool, but very convenient when we have many stock solutions to prepare in a matter of time. It's possible to export and print all this information and to save it in the campaign, so you don't need to go and restart every time. So what we have tried to do through Ilaka is really actually to make a workflow that is close to the workflow that the chemists will try to make in the lab, and to make it very user-friendly. So we are always working on the UI and trying to make to get feedback from what we're working on in the lab and how we can improve this experience inside of the robots of Ilaka. So when you're finished with the campaign, you can complete and put back the information that was recorded by the robot. And it's possible to prepare actually a file where you have the data ready for making LCMS analysis. So we already pre-calculate M plus H and other ions. And this is actually a list directly able to submit to the LCMS analysis department. And we can also make now a solubility report when we are trying to actually gather information of how compounds will be soluble or not at the end of the reactions. So we can try to attribute sometimes if the failure will be dependent on the solubility. So overall, we have a functional robot now with a functional software that is actually combined of several AI layers. And this helps us to make a very integrated workflow approach. And as you can see, coming soon, we will have the integration of the analyticals and LCMS analysis reporting directly inside of Ilaka.
Thank you very much, Frank, Quentin, for the really interesting webinar. Oh, sorry.
I think we still have some.
Yeah, we have some slides. Sorry for that.
Yeah, and those slides actually answer the question about purification and analysis. And I think it's an important topic because where we are now, and what is showing his screen is that, as you can see, we have Mica to generate molecules, Fire for the retrosynthesis, Ilaka, Orchestration AI, and the robots. And the goal now, of course, is to integrate purification analysis and biological testing, which are now outsourced to external partners. And for that, we have applied to a very competitive ground called EIC. It's a European fund, which which is very, very competitive. In total, 1,200 participants, and they selected only 70 projects. So it's really honor us to and the quality of our science and technology. And we have been granted to 2.5 million euros to integrate purification analysis, and also they will participate for the next fundraising round. So if you want to test the platform, it's very simple. We can start, progressively, where you share a list of molecules. We run Ilaka, we show you how many molecules we can make, the one we cannot, and then we can go until we buy the starting material and run the chemistry on the robot. And if you are convinced, we can enter into more advanced collaboration framework on the next slide, where we can customize and integrate our AI, GenAI, retrosynthesis, orchestration to your environment with your hardware. So no need to buy a new robot if you already have some hardware. We are working with other company to integrate on your premises our technology. We can also start from scratch a platform and totally replicate Ictos robotic platform to your premises, or we can also co-develop something new together. So thanks a lot, and we are more than happy to take questions.
Okay, thanks very much, Frank, Content, for the presentation. Now we jump to our Q&A section. We have a few questions already, so we will be reading them in order of submission. We will start with a question from Fabricio that he is asking for the early stages of the process. What is the advantage of the novo using building blocks from fixed libraries compared to exploring, for example, the enamine real space with a fitness function?
Yeah, so the enamine real space is something very interesting, but it's a fixed library. You have billions of components and you can just filter them using your fitness function. The advantage of the de novo design is that you can do anything you have in mind. You can start from an advanced intermediate and grow to a certain direction, to grow your molecules directly inside the platform inside the protein in less than 10, 20,000 docking. You identify something new, feasible, and optimal. No need to screen hundreds of millions of billions of compounds. You can also start from two right-hand side, right-hand side, and left-hand side to find something new in the middle. So you can do macrocyclization. You can impose the chemistry, impose the price. So you have much more liberty and freedom to operate rather than a static library. You can just filter and cross finger that you're going to get something interesting.
Many thanks, Content, for the answer. So now we have two questions that relate to the analytics workflow, part of the workflow. I will read the question from Mark. Could you say something about how you might have integrated analytics into the platform, specifically LCMS, purification, mass direct LC? I know you mentioned it's upcoming, but maybe you can share a bit more on what will be the approach.
Yes, so the approach is actually to buy new hardwares. So we will be able to have obviously HPLC purification machines. And we also have in mind to order a new liquid handler. So we will be able to actually keep our robot only for chemistry, the ChemSpeed robot, and have another robot that is actually preparing everything for purification and analysis. It's really difficult to have analysis in line directly in the ChemSpeed robot because we are running many reactions at the same time that are super different. And you don't want to stop all the time the agitation or using your liquid handler just to make this analysis. So inside of the ChemSpeed platform, we will not make analysis in line, but externally at the end of the process.
Thanks, Frank, for the answer. So Dominique also asked about purification, but maybe you can comment something about how it will be integrated to create the closed loop. So his question is, do you plan to have automated approach as well with further reaction optimization boosted by AI to work in closed loop later on?
Yeah, so obviously this is something that we are having in mind. However, what we are working at Ictos is to have this clustering approach where we just want to have the very general reaction condition. So we are not looking for the best yield of each steps, but more than having the product every time at the end. So it's a different way of thinking of reaction optimization, but we are trying to put models, and anyway, we are recording all the data. And in the future, we'll try to see how we can embed this in ILACA.
Thanks, Frank. So we move to questions from Craig. You show that the robotic platform is typically applied in the heat validation expansion phase, and applicability reduces as the project matures. I am curious to know what the root cause of this reduction is. And is it due to the complexity or length of the chemical route, or the scale of the reactions, or the lack of efficiency to put on the robot for smaller numbers of compounds?
I think it's a mix of everything. So usually when you start a project, you want to go very fast with 1 or 2 steps maximum to explore rapidly the chemical space at low cost and at high speed. And then the more you progress, the more you start performing some tiny modification when you enter in heat to lead. And so the more you complexify the structures, you complexify the position into that structures you want to modify, the more, of course, you increase the length of the routes, and it becomes more and more difficult to use the robot. So again, we really believe that the robot is very, very efficient for heat discovery, heat to lead, and the more you complexify things, it reduces the power of the robot. But still, at some point, even in, let's say, lead optimization, you can prepare on the bench your key intermediates, and then you can assemble them at the end on the robot. So still, we can introduce the robot at some point, but of course, less than compared to the beginning of the project.
Thanks, Quentin, for the answer. The next one is a comment I wrote at Presence at Biotech X. So it's good to mention that for those in Europe, our Chief Business Officer and our European Business Director will be present at Biotech X coming in October. So thanks, Nicolas, for asking. And then we move to a question that has been submitted in Japanese. And thank you to Hideyoshi Fuji, our Business Director in Japan, who provided the translation. So the question is it possible to integrate information related to synthesis, such as reaction conditions, raw material barcodes and loads, and products with external applications like electronic notebooks other than Ictos?
Yeah,
Yes.
sure. Yeah,
yes, of course, we can plug any information actually inside, and I think it's very flexible what we can do with ILACA.
Thank you very much. So we have a few more questions. There is a question concerning the hardware component of the process. So do you think this orchestration and automated synthesis can be adapted to other platforms at camp speed? And if you can comment on this.
Yes, so as mentioned earlier, it's not hardware agnostic. So it's possible to apply it probably to any type of hardware. We just need obviously to take a bit of time for understanding the constraints of each hardware. But we started with camp speed because it was a proximity for choice, for example. They were based in Switzerland, and this is a well-known brand of automated synthesis platform. But we believe that it's possible to apply it to other type of hardware.
Thank you. Another question is around if you can pilot several robots with ILACA.
So yeah, what we have tried to build is a scalable approach where our workflow can be really applicable to other robots. And ILACA on its own can actually drive different way of putting the workload. So we can select within ILACA which robot we want to use. So far we have only one, but we already make some POC, a virtual POC, where we can possibly connect more than only one robot. And this is just another scheduling layer to add.
Thank you very much, Frank, for the answer. So now with this, we reach the time for the webinar. Thank you to the attendees, please, you will find in the chat our contact link if you would like to reach out to the team. You will receive a link with the recording webinar. And we thank you for joining us today. And thanks to our speakers, Quentin and Frank.
Thank you all.
Thank you very much.
Eleonore. Bye-bye.
Bye, everybody.
Goodbye.